Welcome back, everyone, to Behind Our Science. This is episode 5. Hope you enjoyed our last episode 4, that was live, recorded with different guests that talked to us about COVID blues and mental health. For this episode, we have a giveaway. We're giving away 2 mugs. So the mechanism is just tweet at us saying, I watched episode 5 via Twitter, and then you'll get into a raffle to get one of those mugs. In this episode, we're highlighting the day in the life of 2 important guests. The first guest is Dr. Paul Munga. He is an important member of the American Society of Investigative Pathology. He is the director of the Experimental Pathology Program here at the University of Pittsburgh, as well as the head of the PLRC at the University of Pittsburgh. We spoke to him about his career trajectory, his day in the life, and how to do a day every day by day as a chair of a department at an academic institution. As well as we spoke to Abe Beyer from the University of Tufts. He's doing an MDPhD, and we talked to him directly about non-traditional career trajectories, specifically at this times. We also have a specific segment on how to say the American Society of Investigative Pathology. It's at ASIP. We also have an important segment from Daisy Chu. She has a segment on 7.3 questions, which is inspired by Bo's 73 questions. And we also have highlights from the American Journal of Pathology and the HCS by one of our co-sponsors and co-presentators, Vig Meadows. Hope you enjoyed episode 5, and always remember to always believe what's behind our science.
I'm an experimental pathologist. I do treadmill half an hour in the morning and half an hour in the evening. That's just my routine. I have a microscope that sits right on my desk, and there's a teaching head. There's actually a TV that's connected to it. So a lot of the individuals that I'm going to be meeting, we are going to go over slides. It could be graduate students or postdocs from the lab. It could be collaborators who are coming from Children's Hospital or some other places. And we are looking at the slides and trying to interpret some of these lesions, etc., especially, liver. And, being a liver guy, I know exactly what to look for and help our collaborators with that particular part of the study.
And does that involve any trainees? Do you do any training with them under the scopes?
Absolutely.
Are they involved in some of the design and everything that happens with it?
That is exactly right. So a lot of the times, all of this is decided, in lab meetings. I have once a week lab meetings, and unfortunately they're again on virtual Zoom, but we have a robust group of about 14-15 individuals who all join. It's usually on Thursdays. That's where we really decide what the next week is going to look like, as well as discuss some of the progress that was made the week before. These graduate students would do staining on these slides. They would be looking at some specific markers, and then in addition to those weekly group meetings, I meet individually with my trainees, especially my graduate students.
Yeah.
And my junior faculty, honestly, who are in the lab. And we meet and we look at the slides, we make interpretation. There's a lot of learning, there's a lot of teaching that goes on, during those times. So it's really fun. Love to take pictures on my microscope, and I'm the one who designs a lot of the figures for my manuscripts. That's one of the...
So your involvement is also on some of that aspect.
That is correct. I really enjoy doing that.
Is there any non-scientific reading that you mostly like?
Right. So I wish, I had a good answer for that, but unfortunately, no. Other than all the stuff that is, you follow politics, you follow things that are going on, but other than that, I spend most of the time reading scientific stuff. Yeah.
Perfect.
All right. So my day typically ends anytime between 4 and 6, depending on one day to the other. When I'm not stopping for any grocery run, I'm just kind of heading home. And if I'm deciding to go a little bit early, which is at around 4 o'clock, my goal is to get home, again, catch up on emails, because believe it or not, those things never stop coming. You do that, or if I have already thought in my mind a specific task that can take 30 to 45 minutes to an hour to finish, I go home, and straight away get to that, wrap that up, and then it's my time for my second set of treadmill. I do treadmill half an hour in the morning and half an hour in the evening. That's just my routine. So I do that, watch, again, my favorite shows. They're just continuation of what I've been watching in the morning. And then, I'm done. I feel relieved. Come back and it's time to start cooking. I'm a cook. Yeah. So most of the cooking that I do is on the weekend. That's been going on for the last almost 10 years, if I can remember. But on Sunday from 2 o'clock to 4 o'clock is my cooking. That I cook about 5 or so dishes. This is when the kids were young. My wife is a physician as well, so she has a very busy schedule. So we cannot really spend too much time cooking when we come back in the afternoons or evenings from work. So the best thing is to kind of cook, and I love to cook, all sorts of Indian foods, a lot of beans, a lot of Indian tofu and Indian cheese. But at the same time, love my chicken curries. We cook spaghetti, meatballs, all sorts of things.
What's the dish that your family lives without? Could not live without.
I would say tandoori chicken is what I do. So we just kind of marinate the chicken for a day or so, and then the next day I have a special secret sauce that I make, and everybody loves it. So okay. So that's what we've been. Any other doing. Type of foods that you normally go for? Yeah, I mean. Rice,
Italian, Mexican. Is there something?
Absolutely. I mean, literally, the second favorite, because our foods were driven by what our kids loved. So my wife and I would eat what the kids would eat, yeah. and their favorite foods were pizza growing up. Their favorite was spaghetti and meatballs. It's interesting. I have a 25-year-old who's in a medical school, and my son who's an undergrad at UCSD. That since they have gone to their colleges, they feel that they love Indian food more now than when they were growing up. So when they come back, every couple of months, it's always that they like to eat the same Indian stuff that I would cook when they were growing up, which is I don't know how to. Some of those are Indian names, but paneer tikka masala, chicken tikka masala, tandoori chicken. I mean, those are the kind of things that they really love. So, and I can do them really well. So hopefully, we'll do it for you guys someday. Yeah. So yeah, when I and the other thing, and I can blame my son for it, is that he's gotten us completely addicted on salads. He would not eat any meal without a salad, and now we cannot eat any meal without salad, which is great. So when I come up from the treadmill, freshen up, and then my wife is almost on her way back. She'd always call me, let me know that she's on her way, but that's kind of signal that get the salad ready, and that's what I do. So I get my salad ready and take one dish out, from the refrigerator that I cooked on the Sunday before, and that's once she comes, she may decide to do an exercise as well. Then I'll just wait around, either work or just turn on the TV, and watch a little bit of that. And when we always like to eat one meal of the day together, and that's our dinner. So just have dinner, and usually the best time is anytime between 6:30 to 7:30. If we can finish our dinner between that time, that's great. While we are watching, we are again talking about things, but both my wife and I would like we like to watch TV for an hour or so after that. So you can tell that our household is addicted to watching these shows, but we kind of, that's the only thing that sort of unwinds us, helps us relax, and and of course we talk about things, and right after that, the sort of the last major order of the day before we again start working, both of us, is to have a cup of tea together. So, we have a cup of tea. It's, and once the tea is ready, I think that's when the TV is turned off, and now in our house we have like a nice warm fireplace. And we have put a small desk on either side. It's a computer table, which is sitting on both sides of the fireplace. So my wife sits on one side, I sit on the other. Both of us have laptops. She has dictations to use, to do, edits to make, orders to put. I have grants to write, papers to review. And, so that process kind of continues. So the second phase of work, usually starts around, I would say 9:15. And then, we can be up until 11:30, we can be up until 12:30, we can be up until 1:30. So my wife usually ends up, sleeping at about 10:30, 11 for the most part, but I really get active at that time, and I work for another at least 2 hours or so. Really enjoy the science that we do. That's what keeps me grounded. That's home. That's my hobby. What we are working on in the lab is trying to understand some of the cellular and molecular underpinnings of certain types of phenomena that are unique to the liver. For example, the liver's capacity to grow after it's cut and removed. But if you can remove half of it, 2/3 of it, how can that remaining 1/3 grow back? That's an enigma. There's a lot of molecules that we and others have identified. So our contribution to the field has been the cell molecule circuit of the Wnt signaling pathway, and that still continues to. We always are finding something new in that field. And the dark side of regeneration, which is cancer in the liver. So we are also interested in that, and that's an active area of investigation. The other aspect that we are really intrigued by is how liver first develops in an embryo. So those signals, and what are the signals, and what are the cells that are involved in the process of hepatocyte differentiation, bile duct formation, morphogenesis. So a lot of that work is ongoing in the lab. ASIP has been also home. Been working with ASIP since I came as a postdoc back in 1999. As I was beginning to find myself and got my first faculty appointment as a research assistant professor in 2001, I wanted to be more involved with the society, just to kind of get to know people, and that's when I was introduced to Mark Sobel, who was the executive officer. And then when I told him that I want to be involved with the society, he held me by my hand and took me to Bill Coleman, who was then program director, if I remember correctly. And then since that day, I have been involved with the society and been program director. Currently, I'm the secretary, treasurer, and a council member. So, an opportunity for me to give back, to the society. One of the leaders for the special interest group on liver, along with Dr. Kerry Nijek-Bowen. ASIP, I think, it has been instrumental in where I am today. One of the things that is very important when it comes to promotions of young faculty is their service portfolio, component of their portfolio. So you have to have research, you have to have teaching, and you have to have service. If you're a clinician, then you have to have your clinical component as well. But in the service component, being part of a national society, contributing positively to that society, either as a committee member or a part, or directing an initiative for a society, goes a long way. If it was not for ASIP, I wouldn't have been where I am today.
Bayer, like the aspirin, even though there's no relation, unfortunately. I grew up in Connecticut my whole life, and then was also a Tufts undergraduate and have been in the Boston area ever since, so New England through and through. I worked in a chemical biology lab in undergrad, which I liked a lot. I learned how to do research. Some of it, of course, was busy work, learning how to do very basic things, but I wasn't as thrilled with that because it was very basic science, no translational disease applications. So then, before starting medical school, I also worked in a lab at MGH, which was much more translational. It was using mouse models studying iron regulation, and I learned more of the biologic aspects of research, but was not particularly interested in iron as a research field. So then I kind of narrowed my research focus to cancer and started looking at immunology as well. So when I was looking for labs to join for my PhD work, I was introduced to Pilar, who, while she didn't have any cancer projects strongly going at that point, was definitely a strong translational immunology lab and was very open to students coming in with different interests. So it worked out very well for me to come in and get to learn a lot about immunology and start some new projects in my own interests. Pilar. One of the most exciting moments in science in undergraduate was when I had spent the better part of a semester or two optimizing an experiment for my undergraduate lab, and I was sitting at a computer waiting for a Western blot to develop, and all of a sudden it popped up and had the exact result that we needed to prove that this whole project we'd been working on for years actually worked. And I remember quite literally jumping up and down with excitement at the computer, and the graduate student who I was working with ran over and was so excited, and the two of us just sat there looking at a little band on a Western blot, being like, wow, our project actually works. This is wonderful. It is currently actually about to be submitted to Nature Chem Bio at the manuscript, so hoping for good news on that front. At really random times, actually. There have been times when I've woken up in the morning and not remembered a dream, but all of a sudden had an idea for an experiment, or sitting making dinner and all of a sudden I'm accidentally thinking about science and have to stop what I'm doing and go write something down. I don't have a particular place I go to think or a particular time where inspiration happens most, but at random times I have to stop what I'm doing to make sure I don't forget my good ideas. Before deciding to do an MD-PhD, I had decided a lot earlier that I wanted to go to medical school, and I think my hesitation for MD-PhD was that a lot of the MD-PhDs I knew basically had to kind of pick one side of their career or the other. They really had to focus on medicine to be successful, or if they wanted a successful lab, couldn't practice medicine well. So that was my hesitation for that, but I met a handful of MD-PhDs who did it all. There was specifically my hematology oncology professor for the medical school who ran the department, saw patients, had a successful research lab, taught, and had a family, and somehow did all of those things and still was a competent person. So when I realized that there were more people who could balance it all, and then I decided that MD-PhD was the route that I wanted to go, and I definitely would have decided that sooner had I known that it was possible without sacrificing any of them. So I had originally wanted to focus on cancer research when I started and when I was rotating, and something that I had been told was, everyone's working on cancer, you shouldn't do the type of research that everyone's doing because you won't be able to be successful in it. And that, while I do have a project in cancer, I definitely stopped looking at cancer labs in part because I was discouraged from that and was told to kind of try a field of research that was a little less populated. And I'm very happy in the lab that I'm in and happy that I do have some projects on that, but I think I wish I had known that you don't have to listen to that. You can work on things that are very popular and still be successful. Can I share something much more personal? This ended up working out in the end, but when Gavin, my boyfriend, moved out of state, we had been dating for a year and a half, we decided to break up. We didn't want to do long distance, and the 2 or so months we were broken up were probably the worst months of my life. But we decided to break up, we decided to call it off, and ended up thinking that was a very, very poor choice afterwards, and was very upset for a long time. And in the end, we did get back together and are still together, but I think a sort of cliche lesson from that is to not give up when it seems very hard and seems like it can't work, because I wish we hadn't done that. I wish that we had just kept going, and obviously for me it ended up okay, but I could have avoided a lot of pain had I decided to just give it a shot. Like, I was definitely worried about trying to fit in what a long distance relationship would look like and trying to do all of that, because I definitely wouldn't want to sacrifice any sort of success in my career for a relationship that, while it was very strong and important, a year and a half isn't being married for 5 years, so I wasn't, ready to try and sacrifice anything. So it was definitely a worry at first, but then I think once we started trying it again, I learned how to balance and learned that there was some give and take, but you still could manage it. And obviously our relationship looks a lot different now than it did when we were in the same city, but that doesn't mean it's not worth having. As far as MDPhD specifically, I would give the advice to not think of it as 8 to 9 years of school, because each component of the program is so different, and some ports feel like a job, not just sitting and studying. So, you are studying a lot for 2 years, but then your PhD, you really are working a lot of the time, and it feels very, very different than what medical school was like. And then the last 2 years of medical school, you're in the hospital every day. You're functionally considered part of the medical team. So to anyone who says, oh, I don't want to go back to school for 8 years, but if you're interested in this path, it's not sitting in a classroom and taking tests for 8 years. So in the end, it definitely is worth the length of years. So I would say my advice is to look at the program in blocks that look very different and don't feel like studying the whole time. So most days I am in lab, so on one of those days I would wake up and I eat breakfast every day. I go into lab. I am in lab for 8 to 9 hours on a typical day, but in between there could be a 2-hour lab meeting or a 2-hour class or various other meetings throughout the day, so not standing at the bench that entire time. The best resources that have helped me both in medical school and in PhD or research are various students that are years above me. I think I've gotten the most useful advice from older medical students for studying and how to go about every part of medical school, as well as learned most of what I know in lab from people who are more experienced than me. So in my PhD, I've definitely been inspired and learned the most from my lab mates, such as Marina, Sasha, Fran, and Kuljit. I think it's essential to have a good mentor during the PhD years of MD PhD or any PhD, someone who both can teach you and help you become a scientist, but also someone who will challenge you and force you to do some of the thinking on your own to learn.
Daisy. Hi, Manuela. Thanks for being on 7.3 Questions with Behind Our Science.
Thank you for having me.
So what's keeping you busy these days?
I'm mostly working on experiments. I'm doing qPCRs, data analysis, and writing. So that's definitely it definitely is keeping me busy and taking my time.
What's the best vacation you've ever taken?
I think that would have to be going on a trip with my boyfriend to Orlando. That was really exciting, and we got to see Volcano Bay and everything you see in the movies.
What are you currently reading?
I'm currently reading The Vanishing Half, which is taking me a little bit of time to get into, but I do recommend it for anyone who's not an avid reader but is interested in, doing some pleasure reading or reading in their leisure time.
Ooh, what do you have going on right here?
So this is a BCA assay that I'm working on, and yeah, colors are really pretty, and that's one of the things I love about it.
What's your favorite assay?
My favorite assay, while I love this, would have to be the ELISA, because it's really cool to see media that could have been chucked out or trashed, secrete things that we could actually use to tell so much about the cells that we are working on.
And what's your favorite pipette?
Good question. I think the P1000. Mostly because it goes from 100 to 1,000 and you can go crazy with it.
And -80 or -20?
Good question. I think that's really interesting because I didn't know about the -80s before I joined the research lab. So that was really cool to see how cold and frozen things can get in this region. So I have to say the -80.
And what's next on the horizon for you?
The big question. This fall I'm starting a PhD program, or I'm joining a PhD program at Cold Spring Harbor Laboratory in Long Island, New York. And yeah, I'm going to be studying things from cancer to Alzheimer's research, and I'm really excited about that.
Cool. Thanks so much for joining us today.
Thank you for having me. Bye, Daisy.
My favorite piece of equipment is the biosafety cabinet.
Favorite equipment is the ice maker because it's like keeping me cold.
Everyone, my favorite piece of equipment in the lab is the multi-channel pipette.
My favorite piece of equipment in the lab is parafilm.
Hi, I'm introducing my favorite equipment, and this is EVOS. I use this to image my cells, live cells, and also the red blood platform.
My favorite thing in the lab is the Mr. Freeze.
So my favorite thing in lab is this equipment, and for this I can do lots of animal experiment for my experiment.
My favorite instrument is the nanodrop. It's very useful in measuring protein and DNA concentrations.
Favorite piece of equipment is the gloves.
So my favorite tool in the lab would be the P1000. It's essential for allowing us to output and dispense precise amounts of liquid.
So how do you pronounce that?
ASIP.
ASIP.
So how do you say it?
It's ASIP.
How do you pronounce ASIP?
I pronounce it ASIP.
So how do you pronounce this?
ASIP.
ASIP, right?
ASIP.
ASIP.
Of course, it's ASIP.
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 ASAP.
ASIP. ASIP.
Did you know that artificial intelligence can help you care for your kidneys? As Dr. Jiang shows in the August issue of AJP, glomeruli, the tiny network of blood vessels that work as the cleaning units of your kidneys, can become damaged and lead to disease and kidney failure. Routine staining and analysis done by pathologists are common for proper diagnosis. However, the authors were able to build an automated program utilizing stained images and scans from over 600 patients that could classify multi-stained glomeruli with efficiency and robustness, meaning it can be applied as a first step for detailed kidney histologic analysis, leading to increased efficiency for both patients and doctors alike. Check out this phenomenal paper online at AJP. Proteins are expressed by genes, but what regulates their expression? That's where specialized proteins called transcription factors come into play. Both in health and disease, they regulate the expression of proteins necessary for cell survival. Dr. Caprio and co-authors used immunohistochemistry, a way to detect proteins in tissue slides, and computational analysis of online databases to analyze gene expression of CREM and its fusions across human tissues. They found that normal CREM protein is widely expressed in almost all normal human tissues and cancer tissues. However, their data showed that CREM expression can also be fused with other proteins in tumors. The wide expression of CREM protein in normal human tissues and cancer limits the utility of immunohistochemistry for identification of tumors with CREM. However, these findings are important for the advancement of cancer studies and detection of cancer biomarkers. Check out this open access article online at the Journal of Histochemistry and Cytochemistry.
Thanks for watching, everyone. Don't forget to tune in for our live episode in September. That will be episode 6 of Behind Our Science, the podcast. For episode 6, we will be celebrating Hispanic Heritage Month. Don't forget to subscribe to our YouTube channel and be on the lookout for when we upload the individual segments from all our interviews and our featured guests. And don't forget, this is Behind Our Science.